Hello everyone, welcome back to Bioschool. This is Gayatri. So today's session we are going to discuss about transduction. Transduction is a type of horizontal gene transfer. In my last two videos I have discussed about transformation and conjugation. So I will drop the link in the description box down below. So please make sure to check that video. So let's get started and understand what is transduction and how gene is transferred. So what is transduction? Transduction is a process of transfer of genetic materials between two bacteria via virus. Transfer of genetic material from one bacteria, let's say this is one bacteria, and the gene is transferred from one bacteria to the other bacteria by a virus. So the transfer of genetic material from one bacteria, let's say the donor bacteria is bacteria A to the recipient bacteria, and the recipient bacteria is B. So the genetic material is transferred from one bacteria to other bacteria via virus. Okay, and this virus, it is known as the bacteriophage. Okay, the virus that infect the bacteria is known as bacteriophage. Okay, so the transfer of genetic material in bacteria, that is from donor bacteria to the recipient bacteria by using a virus, or the virus that infect the bacteria is known as the bacteriophage, is known as transduction. You know, the transduction are of two types. What are those? One is known as generalized transduction. First one is known as generalized transduction. So let's discuss about generalized transduction first. Why it is called as generalized transduction, I will tell you later. When the bacteriophage, it infects the bacteria, let's say this one is the bacteriophage. When this bacteriophage, it infects the bacteria, then either it will undergo lytic cycle, or it may go lysogenic cycle, right? So when the bacteriophage, it undergoes lytic cycle, then generalized transduction, it occurs. Okay, in lytic cycle you will find generalized transduction. And if the bacteriophage, it undergoes lysogenic cycle, then in the lysogenic cycle you will find specialized transduction. Okay, let's have a look on more details about both the generalized and specialized transduction. Okay, so this one is the bacteriophage and this one is the bacteria. Okay, this one is the donor bacteria. So when the phage, it infect the bacteria, then the phage DNA, it will enter into the bacterial, right? So this one is the bacteria and this is the bacterial DNA. So when the phage, it infects the bacteria, then the phage DNA, it will enter into bacteria. Okay, phage DNA, it enters into the bacteria, and the empty phage coat, it remains outside of the bacteria. Okay, in the next step, when the phage, it infects the host cell, then it causes the degradation of host DNA into small fragment. Okay, so when the phage DNA, it enter into the donor bacteria or the host cell, then what will it do? Then it will take over the host machinery and it will degrade the host DNA into small fragment like this. Okay, then in the next step, so what happened here? The degradation of host DNA. Then in the next step is the phage, it take over the host machinery. So now the phage coat proteins are synthesized and phage DNA is also replicated. Phage coat protein are synthesized as well as phage DNA is also replicated. So there are the many copies of phage DNA, and these are the host, bacterial host DNA. So in the third step, what happened? Phage coat synthesized and phage DNA replicated. Then go for the next step. In the next step, during the process of maturation, of the virus particle, a few phase head, they contains the bacterial DNA instead of phase DNA. Okay, in the process of maturation, of virus particles, the few phase heads, okay, these are the phase coat or the phase heads, they contains the bacterial DNA instead of phase DNA. Let's say this one, it contains the bacterial DNA, I have instead of phase DNA. The red one is the phase DNA and the blue one is the bacterial DNA. So here some phase head, not all, some phase head contains the bacterial DNA, some phase head it contains bacterial DNA instead of viral DNA. So in the next step, what will happen? This is another bacteria. If it is the bacteria 1, let's say this one is the bacteria 1 and this is the bacteria number 2. Okay, bacteria 2, I represent the DNA in yellow color. So if it is infected by this bacteriophage which contains the bacterial DNA, okay, it contains the bacterial DNA or the DNA of bacteria 1. Okay, mostly the bacteriophage, they contains the viral DNA, but here it contains the bacterial DNA. If it infect the host, then what will happen? This is the bacterial DNA and is the bacteriophage that contains the bacterial DNA of one bacteria, it infect the another bacteria. So this bacterial DNA, it will enter into the new host cell and become integrated into the bacterial chromosome, thereby transferring the several bacterial genes at one time. Okay, so as it enter into the bacterial DNA, then in the next step what will happen? It will integrated into the bacterial chromosome, thereby transferring the bacterial gene. Okay, so here you can see the gene or the bacterial gene, it is transformed from one bacteria to another bacteria via virus. Okay, via virus is known as transduction. When the DNA of bacteria 1 it enter into the bacteria 2, then the DNA it will integrated into the second bacteria's chromosome, right? Either it will integrated or it may be disintegrated. Okay, that means the transduction it will be successful or it may be unsuccessful. Okay, or there is no success like that of transformation as I have described. So if I say that what is the difference between the transformation and transduction? So in the transformation, the naked DNA from the environment which is taken up by the bacteria. Okay, but here the DNA is transformed via a bacteriophage. Okay, so this is the first difference between transformation and transduction. And in the transformation, only the single stranded DNA is transformed from one bacteria to another bacteria. But in transduction, okay, in transduction, this is the double stranded DNA. Okay, in transduction, double stranded DNA is transformed from one bacteria to another bacteria. So now the question is, why it is called as generalized transduction? Because the donor DNA from any region of the chromosome have chance to enter into another bacteria. Okay, let's say this one is the donor bacteria, right? So any region of the chromosome have chance to enter into another bacteria. It may be this fragment, it may be this fragment, any fragment of donor DNA or the bacterial chromosome have chance to enter into the other bacteria. That's why it is called as generalized. There is no specialized region of bacterial DNA is transformed to other bacteria. So in generalized transduction, any portion of bacterial chromosome can enter into another bacteria. Okay, or it can be transferred, or we can say that any region of bacterial chromosome can transform to another bacteria via bacteriophage is known as generalized transduction. Okay. So this is all about generalized transduction. So let's have a look on specialized transduction and why it is called as specialized transduction. So as I have told you here that the specialized transduction we can find when the bacteriophage it undergoes lysogenic cycle. Okay. So this one is the bacteria and it contains the bacterial chromosome, and this one is the virus or the bacteriophage. So when the bacteriophage it infects the bacteria, then what will happen in the next step? The phage DNA it integrates into the bacterial chromosome. It integrates into the bacterial chromosome. But in the generalized transduction, it has not integrated into the bacterial chromosome. Okay, so here it will first integrated into the bacterial chromosome. Then in the next step, during the disintegration, pick the sum of the bacterial chromosome. Okay, then it will go for the next step. In the next step, replication of viral DNA and destruction of bacterial DNA. Okay, destruction of bacterial DNA. Bacterial DNA it is disintegrated, and the viral DNA it will replicate, and the viral DNA it also contains the portion of bacterial chromosome. It will be replicated. So what will happen here? Replication of viral DNA and destruction of bacterial DNA. Then in the next step, the viral capsid or the viral head it will synthesize and it will contain the viral DNA. So here the viral DNA is present, but the viral DNA it also contains some portion of bacterial DNA, right? Like this. Then because of the some stimulation, it may be by the chemicals or by UV light, then the lysis of bacterial cell takes place and the phage it is released, and then they will infect a new bacterial cell. Okay, right? When this phage it infect another bacteria, let's say the second bacteria, it has yellow chromosome and it is infected by this bacteriophage. Okay, this bacteriophage it infects this bacteria. So now in the second bacteria has its own DNA. Second bacteria has its own DNA. Also the DNA of first bacteria, the blue one, the DNA of first bacteria along with some viral DNA. Okay, red one is the viral DNA. So in the next step what will happen? The bacterial chromosome, if there is a homologous region, then they will recombine and the viral DNA it will remain outside. So this is the process of specialized transduction. So specialized transduction, why it is called as specialized transduction? Because here the bacteriophage it transfer only specialized gene or the restricted gene from donor bacteria to recipient bacteria. Okay, like so here the bacterial DNA which is present near to the integration site. Okay, near to the integration site of viral DNA is only transferred via bacteriophage to second bacteria. That's why it is called as specialized transduction. In generalized transduction, any portion of the bacteria, maybe this side, that's this side, maybe here, maybe here, any portion of the bacterial chromosome that is transferred to another bacteria. But in specialized transduction, only the portion of bacterial region that is present near to the site of integration will be transferred to the second bacteria via bacteriophage. That's why it is called as specialized transduction. Okay. So in transduction, only the genes that are located close to each other will be transduced together. Okay, let me write down here, which is also called as co-transduction. As I have told you in transformation, there is a co-transformation. Similarly, here it is co-transduction. Co-transduction means the genes which are located close to each other, they will be transduced together. Okay. So this one is the very important point. So this is all about transduction. In the next session, I will solve some previous year CSIR-NET questions from transduction. So guys, please stay connected and bye bye. See you in my next video. Till then, keep learning.